Hello, good evening to all of you. Good evening, Nidhi. I am audible to you. Please let me know my voice is audible. Are you able to hear me? Any one of you, please let me know. Okay, okay. So yes, a very good evening to all of you. Hope you all are fine and doing great in your own life. And how are you all? Everything is fine. Please let me know. I'm just watching your assignments right now. Some of you have made so many beautiful diagrams, well labeled, neat and clean. Very good. Shivangi's assignment is here. So you all are making it very nicely. But still, only few assignments are here. Like yesterday, I told you to make the assignment of nucleus, the cell, basically the structure of a cell, along with endoplasmic reticulum and Golgi body. But only few assignments are there. 97 members are there in the genetics class, and at least 50 assignments should be here. So only those will get the certificates whose assignments are there. So if you are not able to send me the, if you are not able to attend the classes, it's okay. You may be busy or something in your exams definitely. But at least you have to send me the assignment. So please keep this thing in mind. So at the end you have not to tell me that my certificate is not here. So please keep this thing in mind. Your assignment should be here. From the past 2 3 days I am watching. From 97 only few are there, and I request all of you to send it in a group, so that I am able to keep the record. Otherwise, how I will be able to keep the record? So I request all of you to do the assignment. Why you are not taking it seriously? It will affect you later on. Please, please. I request all of you to share it in the group, and so that when I am just watching the group, when I just opening your assignment, I am just making the record on my book also, on my copy also. So I request all of you be serious. Yes, Shivani, I got it. 97 members are here, but only few of you are the only ones who are taking it seriously. So those will be given the certificates who will be able to send me the assignments as soon as possible. I don't know why they are not taking it seriously. Are you able to get my voice properly? My voice is audible or there is any disturbance? Please let me know my voice is okay. Yeah, Shreya, it's your wish. If you are comfortable on writing on on laptop, then you are most welcome. If you are comfortable in writing it in the book or in a copy, still it's okay. So just no worries, no worries. You can take your time, but at least upload it when you will be free, when you will be able to upload it. Because I have to keep the record of that particular day. Getting my point? So that is why please take it seriously. Okay. And one more thing is that I am not forcing any one of you to, uh, be present there when I will be here taking the classes. No issues. If you are busy, you have your exams, you can open the link anytime when you will be free, and you will get the knowledge at any time. No issues at it all. But you have to submit your assignments. Please, please keep this thing in mind. Okay. So I hope all you get it. After it, I have one more thing to share. So are you getting me? Please reply. All of you, please reply if you are getting me properly. We have Shivani, we have Shreya, Shilpi, Prityasha, Nidhi. I request all of you to reply. Seema, I request all of you to be, because I'm not able to see any of you, so your messages are only the one by which I will get it. Okay, thank you, Sagarika, Shilpi, Nadhan, Abha, Vaishnavi, Nidhi, Shivangi, Aboli. Okay, thank you all. Yes, one more thing which I want to discuss with all of you. As I came to know that many of the students, they have the exam from 15th of December. 15th or maybe 16th of December, because I get so many messages regarding it, that they have their exam from 15th or 16th of December, and they will not be able to, send me the review paper on time or the text on time. It's okay if you will not be able to. Please be focused on your exams first, and after it you have to send me the abstract and topic. Okay. So those who are free, 10th December is the last date for submitting the abstract as well as the topic. So when you will submit everything on time, definitely there will be no issues when we have to allow you the certificates. Because I am just making the record, so I have to check only that for Abhishek Yadav abstract submitted, Sudeshna abstract submitted, topic this one, Shivangi topic this one. So it will not be difficult for me at that time. Okay. So that is why 10th of December is the date, and I suggest all of you to do it if you are free. If you have your exams, then we can't do anything. Then you have to do your exams also. Yes, good evening, Bhavni. And now the another thing which I have to do, which I want to discuss, like we have some topics like we already discussed yesterday, which are quite interrelated. Which are quite interrelated. So what I will do, I will take these topics and I will want to do it on the same day. Suppose if this thing, like this cell organelles is different, like chromosome structure, morphology, number, which we have to do today, and mitosis and meiosis is also related to this thing. So I will try to take the two lectures on a single day, and I will try that we will extend the lecture. Instead of 1 hour, we will try to keep it for 1.5 hours, 1 hour 45 minutes. But I want that we all will be free by 15th or 16th of December, so that you will be free from this lecture series and you will be completely focused on review paper, plus you will be able to do your exams also. So are you comfortable by this or not? Like some topics which are quite related, like this numerical chromosomal abbreviations, polyploidy, structural chromosomal abbreviations. So these two topics are related. So I will try to take these topics together, and I will take the extra time so that we will do it on the same day. So please let me know, are you comfortable in it or not? Are you getting me? Are you comfortable in it or not? So if I will take your 20 to 30 minutes more, are you comfortable? If we will do like it, then we are able to conclude the lecture series by 15th of December rather than 20th of December. Getting my point? Then you will be free for your review paper as well as for your theory exams, your paper, your so many things are there in the college and in institutes. So that is the only thing I want to discuss with all of you, because so many are trying, so many students are messaging me personally that ma'am, we have exam from 16th of December. So is it comfortable to all of you? So because I know some topics are quite interrelated, like this, and this is my nature. When I am doing something and I want to complete it fully, when we are working on a chromosomal abbreviations, then I want that we will complete numerical as well as structural together. So are you comfortable in it? Please, all of you, please reply me. Please reply me. All of you, are you comfortable by this? If we will take a little bit more time, and we will try to yes, good evening, Jeet. How are you? I'm just taking these topics in this genetics lecture series. Basically, I'm just take it today. Also, we have to do the chromosomal morphology, and I just want to take these topics together so that we will be able to do it on a single day. or I will take the extra time, extra hours, so that we will conclude it on a single day, and then next we will move on the next topic. So are you getting it? Because I know many of you have your exams by 16th of December. Are you comfortable, everyone? Please let me know. Or otherwise, you will be able to watch it later also, no worries. But this is my suggestion to all of you. We will take 15 minutes before also, or you can say we will do 15 minutes after also. We will take little bit long session, so that you will able to get it. Is it okay? Are you comfortable? Please reply, all of you, please, please. Shilpi, the timing is fixed. That timing will be by 8 or 7:45 p.m. But be prepared that instead of 1 hour, it will be 1 hour 30 minutes. It will be 1 hour 20 minutes. So be prepared for 20 to 30 minutes extra. Definitely, because if we want to take the long topic, we definitely need more time. So be prepared with 30 minutes more. Only 30 minutes. That's all. Or otherwise, if you will be busy for the last 10 to 20 minutes, you can watch the series again. You can watch to the link again, and you can watch it as soon as you will be free. So I'm not forcing any one of you to be here, but yes, you should complete your assignments. You have to submit it on time, and yeah, when you will be free by 15 to 16 of December, you will have ample of time for your review paper also. Okay, so okay, I think that's enough. Now we have to start the session also. Yeah, so the timing will remain same, but definitely on Sunday and Saturday, as we discussed before also, on Sunday and Saturday it will be a 2 hour long session. If we want to conclude the lecture series, it will be a 2 hour long session on Saturday or maybe on Sunday also. Okay, so be ready with it. So please be free on the weekends, because I want to conclude the lecture series by this weekend. So I hope you all will understand. Yes, the starting time should be 7:45 only. By 7:45 p.m. we will start, and from 7:45 we have to go it by 9 p.m. 9 or 9:15 p.m. So be ready with it. Okay. So let's start now the session. But be free on weekends, please. All of you, please be free on the weekends. I will share the link by 7:30 in the group of you. You have to join by 5 to 10 minutes, because I just take 5 to 10 minutes for interacting with all of you. After it, the class will be start and you have to get online. Okay, when you will be free, just click on the link and be there with me. Okay, so now the tentative timing is from 7:30 to 9. Okay, just for these 1 or 2 days, so that we will able to do little bit more, because you know, like, this is 13 minutes right now, and I am just interacting with all of you. So don't worry, take do your homework which you want to do, and then join the session. So no worries, Shilpi, you just join the session when you will be free. Okay, so now let me tell you all that we have done Mendel's law of inheritance. On the very first day, we have done types of gene interaction, in which we have studied complementary genes, epistasis, supplementary genes, duplicate genes. Okay, so we have done dominant epistasis, recessive epistasis in case of types of gene interaction. The third is quantitative traits and qualitative traits, in which we have studied the Mendel's law, the Mendel's traits which are there, and the another one in which we study the skin color in humans, the eyes color. So we have done those traits also. Multiple alleles we have discussed yesterday, and the examples of multiple alleles are blood group in human, in which IA and IB are able to express its own cells, so they are known as codominant. Okay, then coat color in mice, and one more example is there in case of multiple alleles. After it, I have not taken this lecture 4, lecture 5, and then I start this ultrastructure of cell and cell organelles and their functions. So in this we have just come to know basically what is cell. This is a very basic topic, so I just want to do this first. So we have discussed what is basically cell and what are the organelles which are present in the cell. Many of you have sent me the assignment, but still many are left. I think only from the 96, only 10 or 12 are there who send me their presentations or assignments. Rest are pending. So I will not take it later on. So be ready with it. So in the cell organelles, we have done nucleus, okay, mitochondria, endoplasmic reticulum, Golgi body is there. Everything we have discussed about the cell organelles, and there are some specific functions of that cell organelles. I will share the PPT of all these today. I hope so. I will share because I forget many times. So you have to remind me again and again, ma'am, please share the PPTs, otherwise I always forget. So I will send day 1, day 2, day 3, so I will send all these PPTs, and also I will send the PPTs which we will discuss today. So today we have to study chromosomes. Basically, what are the chromosomes, the morphology, their number, their types, what is karyotype, what is ideogram. Along with it, I want to conclude mitosis and meiosis also. Okay. So now you have to tell me basically what is a chromosome. So this is a question for all of you. What is chromosome basically? Any one of you, what is the chromosome? Please reply. Have you done it before also in your previous classes, maybe in the plus one, plus two, or in the bachelor classes? What is chromosomes basically? Chromosomes. Okay, so chromosome is the condensed form of a DNA. Okay, good. Please reply all of you. What is chromosomes and what is the function heavily packed DNA molecule? Okay. Chromosomes are thread-like located inside the nucleus of animal, and it is made up of protein and single molecule of DNA. Okay, they are in shape of threads. They are small thread-like structure present in the nucleus. Yeah, so I think you all have done, you all are aware regarding the chromosomes. Now this is your homework also. Please write it somewhere because any of you have to upload it in the group also. Now what is the difference between or how DNA, yes, DNA, chromosomes, DNA, chromosomes, and one more term, mm, which I also forget right now. Yeah. So you have to tell me how DNA, chromosome, and genes are related with each other. That means you have to tell me the definition of what is gene. Number one. What is DNA? Number two. What are the chromosomes? So how these three terms are interrelated, you have to tell me regarding it. Chromosomes, genes, and your DNA. So this is your homework. Please write somewhere so that you just share with the students in the group also. Yeah, gene. And now I'm just opening my presentation, but you have to do this homework later on. So let me open the PPT now. So this is your homework only. Otherwise, in the end, we forget always. I forget what we have. Yeah. So the chromosomes are here in the cell and cell organelles, which we have discussed yesterday. So I will share this PPT with all of you. So we have done the history, everything related to cells, the characteristics of cells, the size of cells, the shapes of cells. Then this is the main thing, the structure of the cell. These are the 12 organelles which are present inside the cell. Okay, I'm just going to just do it again. So we have 12 organelles which are present in the animal cell. You must know the functioning of each organelle. You must know the functioning of each and every organelle. What is the functioning of mitochondria? What is the functioning of lysosome? What is the functioning of nucleus? You must know it. Okay. So this is the cell structure which I want all of you to draw. But still many are pending. So this is the nucleus. Everything is here. Similarly, we have a plant cell also. Then we have the bacterial cell also. These are the organelles which are present inside the bacterial cell. Then nucleus, cytoplasm, endoplasmic reticulum is here. A Golgi body is here. So I just give you this homework that you have to tell me how this endoplasmic reticulum, this one, and this Golgi body, how they are interrelated with each other. This is basically your homework. You have to tell me how the processing of everything occurs from this endoplasmic reticulum or from this reticulum up to this Golgi body. That means the functioning of both of these together. Then lysosomes are there, mitochondria is there. Now what is basically mitochondria? Mitochondria is known as the powerhouse of the cell because mitochondria is semi-autonomous in nature and yes, this is what again the another homework which you have to tell me why mitochondria is known as semi-autonomous or why mitochondria is semi-autonomous in nature. This is your second question. So you don't have to reply on the YouTube, no worries. You have to reply in the assignment only. So this is a mitochondria which have the finger-like projection which are known as cristae, and the mitochondria is quite important in the synthesis of ATP, in the synthesis of or the processing which are occurring in our own body. So mitochondria is quite important. So but why it is semi-autonomous in nature? Why? You have to let me know. Similarly, we have plasmids, chloroplasts, we have leucoplasts, we have this is again we have the chloroplast, then we have the centrosomes, cytoskeleton is there. Now what's the difference between prokaryotic and eukaryotic cell? This is very basic, you can do it by your own self. So this is whole thing which we have discussed yesterday. This is the animal cell structure which you have to draw. So I'm not going in detail regarding it now, but I just only let you know that these are the things. So mitochondria is outer membrane, in inner membrane the finger-like projections are known as cristae. Now if you are able to see, this is again the mitochondria, and inside the mitochondria, this is the finger-like projections. Inside the mitochondria, the TCA cycle or the citric acid cycle is there. The respiratory chain oxidation is there inside the mitochondria. We are able to get ATPs. So then we have endoplasmic reticulum. If you are able to see that it is in association with the nucleus. So this is all things which we have done yesterday. Now we have to focus on the today's lecture series, which is on. Now then this is nucleus is there. Now we have to focus on our today's lecture, which is on the chromosomes basically. So I will share this PPT. You will have to go it by your own self. These are just the presentations which we are able to see under the electron microscope. So this is the mitochondria. Okay. We already discussed these things yesterday also. So that's all in case of cell structure. This is the animal cell. This is the plant cell. In the plant cell we have a large vacuole, if you are able to see here. This is animal cell structure. You can go by your own self also. Now today we have to do chromosome structure and function. Okay. So chromosome structure and function we have to discuss it today. So it was in 1875 when a scientist whose name is Strasburger first discovered set-like structures which appear after cell division, not after during. So he was the first who was able to discover thread-like, minute thread-like structures which appear during cell division. Now what is cell division basically? So in cell division, the cell is able to divide. So the cell is able to divide. From one cell we are able to get two cells. Na? So this is known as cell division. So from one cell we are able to get two cells. So we have to discuss cell division right after this lecture. After chromosome we have to start mitosis and meiosis. So Strasburger in 1875 first discovered set-like structure which appeared during cell division. These thread-like structures were called chromosomes due to their affinity for the basic dyes. That means chromo means color. So when we are applying the dyes, these chromosomes are able to give us the color. So the term chromosome is derived from a Greek word chroma. Chroma means color. Soma means body. So when any organelle or anything is able to give us the color, so they are known as the chromosome. This term was first used by Valdier in 1888. Okay, so it was discovered in 1875. Till it take too long time to name it, to term it. So what is the functioning of these chromosomes? Chromosomes contribute to the division of cell. That means from one cell we are getting two cells, and they are of prime importance as they carry the genes. So the chromosomes, you have to keep this thing in mind. Chromosomes carry the genes which are the hereditary material. So the genes are present where? The genes are present on the chromosome. Please keep this thing in mind. This is the important feature, that the chromosomes, they contribute to the division of the cell. From the one cell we are able to get two cells. Along with it, these chromosomes are of prime importance as they carry the genes which are hereditary material. So the genes are present on the chromosome. So number one is chromosome, then comma genes, and then the DNA. So you have to you must know the difference between all of these three, or the basic definition of these three. Now, so this is a chromosome. Now what is chromosome number? So the number of chromosomes in a given species is generally constant. That means that much number of chromosomes are present only. The number is constant. All the members of the species ordinarily have a definite and a generally a constant somatic and gametic chromosome number. So we have two terms. We have two types of cells. Somatic cells as well as the sex cells. So we have two types of chromosomes. We have the somatic chromosomes and we have the gametic chromosomes. So somatic chromosomes number is the number of chromosomes which is present in the somatic cells. As I told you, we have two types of cells. We have the somatic cells and we have the sex cells. So somatic cells and it is represented by 2N. Generally, somatic cells contain two copies of chromosome except the sex chromosome. So both the copies are ordinarily identical in morphology, gene content, and in the gene order. So these somatic cells, the chromosomes which are present in these somatic cells, they are identical in their morphology, in the gene content. So they are known as homologous chromosomes. They are known as homologous chromosomes. So first we have a fixed number of chromosomes. Number one, the chromosomes are present in two types of cells, somatic cells as well as gametic cells. Somatic cells, the chromosomes which are present inside the somatic cells, they have fixed morphology, gene content and gene order. Now what about the second one, which is the gametic chromosomes? Which are there? So gametic chromosome number is exactly half of somatic chromosome number. Getting my point? So if somatic cell is 2N in number, then gametic cells will have N number of chromosomes. Are you able to get it? Somatic cells are generally represented by 2N. So 2N in case of somatic cells. So gametic chromosome number is exactly half of somatic chromosome number and is represented by N. So it denotes the number of chromosomes found in the gametes of a species. So the somatic chromosome number is 2N and gametic chromosome number is N. So if you are able to see here, this is the chromosome number. So if we are saying 2N, then these chromosomes are present in the somatic cells and they are known as somatic chromosomes. Haploid are present in the sex cells and they are known as gametic chromosomes. Similarly, sometimes we have triploid also, tetraploid also. We will discuss these also. But the main one are only these two: diploid chromosome number and the haploid chromosome number. So if you are able to see here, here we have two pink. That means same replica of one. Similarly, here we have two of replica of one. So copy of one basically. Here we have single only. So in triploid we have 3, in tetraploid we have 4. Similarly, if we have 5, and then definitely we will have the 5. But the two main are these only: our gametic chromosomes as well as our somatic chromosomes. Now chromosome size. So the size of chromosomes show a remarkable variation depending upon the stage of cell division. So the chromosome size, it varies according to the cell division. So basically when we will do mitosis and meiosis, then you will able to get it. So longest and thinnest during the interphase is not visible during the under the microscope. These are the stages in case of mitosis and meiosis, which is in interphase, prophase, metaphase, anaphase, telophase. We will discuss there later. Smallest and thickest during mitotic metaphase. So I am not discussing it right here, but in the mitotic and meiosis I will. So chromosome size is not proportional to the number of genes present on the chromosome. So size may be vary. So size is not proportional to the number, that many number of genes are present. So size may be vary. Now this is the chromosome morphology. The outer covering or the sheet of a chromosome is known as pellicle, which encloses a matrix. So outer layer is known as pellicle. Within the matrix lies the chromatin. So inside this we have the chromatin. Fleming introduced the term chromatin in 1879. So Fleming was the one who was able to introduce the term chromatin. So the chromosome morphology and changes during cell division and mitotic metaphase is the most suitable stage for the study of chromosome morphology. So if you want to work on chromosomes, if you want to study the morphology of chromosome, the structures which are there in the chromosome, then you have to study the cell division, because their shape, their size changes during the mitosis and meiosis. Chromosome size, shape, everything changes. So if you want to work on chromosome morphology, or you want to know about chromosomes, so you must know everything related to cell division. So in mitotic metaphase chromosome, the following structures can be seen. So when you are going through metaphase or the this stage, under the microscope, if you are just watching this, like for our students, we have when we are doing the experiment, there is a mitotic stage, and in the mitotic we have the metaphase. So all the students have to do it on the slide and show us the mitotic stage. At that time they will able to see the chromosomes, and even in the chromosomes we have chromatids. So they will be able to understand what is basically chromatids. Like these are known as chromatids, these one. And then centromere, which is there in the center. So in the center we have sorry, I'm not able to draw here. So in the center we have the centromere, telomere, which is on the end basically, secondary constriction, chromomere, chromonema, matrix. So all these are terms which are related to the chromosome. Now what are chromatids? The very important thing. These are the chromatids basically. So each metaphase chromosome appears to be longitudinally divided into two identical parts, each of which is called chromatids. So these are known as chromatids. Each metaphase chromosome, now we are just discussing about the metaphase stage of the mitosis. When we will do that also, you will able to understand more clearly. So each metaphase chromosome appears to be longitudinally divided into two identical parts. Each is known as chromatids. So these are known as sister chromatids when they are present on the they are attached to the centromere. So chromatids of a chromosome appear to join but together by the centromere. So centromere is the one by which the two chromatids are joined. Two chromatids making up a chromosome, they are known as sister chromatids. Now they are making a chromosome. For a single chromosome, two chromatids are involved. So for these two chromatids, they are known as sister chromatids. The chromatids of homologous are known as non-sister chromatids. So now these two are sister chromatids, but when we are comparing this and this, it is known as non-sister chromatids. Getting my point? Because they are present on a different one, na? They are present on a different one, but these two are the sister chromatids. When we are comparing this and this, then they are the non-sister chromatids. I hope you get it. Okay, otherwise this is the homologous pair of a chromosome. So if I will ask you how many chromatids are here right now, so how many chromatids are here? We have 1, 2, 3, 4. So we have 4 chromatids. Okay, so it is basically quad nature, 4 chromatids, 2 sister chromatids and 2 non-sister chromatids. Then the centromere, which is present in the center, if you are able to see. So centromere is the region where two sister chromatids appear to be joined during the mitotic metaphase, and it is known as centromere. It generally appears as a constriction and it is called primary constriction. So where we have the centromere, it present, this is the constriction basically, and it is known as the primary constriction. What is the functioning of the centromere? So it helps in the movement of chromosome to the opposite poles during anaphase of the cell division. So when we will do the anaphase, you will able to know the functioning of centromere. So the centromere consists of two disc-shaped bodies which are known as kinetochores. Normally chromosomes are monocentric, having one centromere each. So generally we have only single centromere which is present in a chromosome, that is why it is known as monocentric. But depending upon the position, they may be metacentric, submetacentric, acrocentric and telocentric. Let me show you a diagram if we have. Okay, so sometimes what happens, instead of presenting just in the center, the centromere may be present at the one point only. That means, like in the metacentric, the centromere is present in the center. In the case of submetacentric, the centromere is located on the one side of the center, such that one arm is long. So what will happen? One arm is long and other arm is short. If it is present in the center, so we have both arms of same size or same length basically, but in submetacentric, one arm is long and one is short. In acrocentric, centromere is located close to the end. In acrocentric, the centromere is just at the end. Similarly, in the telocentric, centromere is located at one end of the chromosome, so that chromosome has only one arm, and they are known as eye-shaped or rod-shaped. So this is your homework. Now you have to draw these 4 types of chromosomes: metacentric chromosome, submetacentric, acrocentric and telocentric. You have to draw these 4 types of chromosomes only. Now what is basically telomere? The two ends of the chromosomes are known as telomere. What is basically telomere? The ends of the chromosome. These are the ends of the chromosomes. They are known as telomeres. They are highly stable and do not fuse or unite with the telomeres. Any broken end of a chromosome is unstable and can join with a piece of any other chromosome, but the telomeres impart stability to the chromosome. So these telomeres provide stability to the chromosome. Then we have another term which is there in the chromosome, which is known as secondary constriction. Now what is secondary constriction? The chromosomes having a secondary constriction, like this is the secondary constriction, this is the secondary constriction, this is the centromere. If you are able to see, these are the two chromatids basically, which are joined here by the centromere. So there is secondary constriction here. That means they are just constrict at certain points. Now what's the functioning of it? The chromosomes having secondary constriction are known as satellite chromosomes or SAT chromosomes. Chromosomes may possess secondary constriction in one or both arms of it. Chromosomal and distal to the secondary constriction is known as satellite. So these, when they are present, when the secondary constriction is there, the chromosomal end is known as satellite. So these are known as satellites. Production of nucleolus is associated with secondary constriction, and therefore it is also called nucleolus organizing region, that is NOR. Satellite chromosomes are often referred to as NOR chromosomes. Are you getting it? Are you here? Let me check it first. because I'm just speaking. Just let me check. Yes, yes, Shivani, you are right, but you have to write the answers later on in the assignment. So up to now, is it clear? Are you getting me properly? Are you getting me? Everything is fine till now? I think it's fine. So let me complete it then. Okay, okay. Thank you, Snadhan. Thank you. Because we have 243 slides and I have to complete it. Yeah. So now we have the chromomere. So this is the primary constriction along which we have chromatids attached. Sometimes there is constriction which is known as secondary constriction, and the end part of the chromosome is known as satellite body or satellite chromosome. So in some species, chromosomes in packaging stage of meiosis show small bead-like structures called chromomeres. Sorry. Oh, sorry. So basically, this will be happen in the packaging stage of meiosis. So I will let you know what are chromomeres there at that time. But we have to keep this thing that we have chromomeres which are there in the packaging stage of meiosis. The distribution of chromomere in the chromosome is highly characteristic and constant. The pattern of distribution being different from different chromosomes. They are clearly visible as dark staining bands in the giant salivary glands chromosomes. Chromomeres are region of tight folded DNA. So if you are able to see here, the chromomere is the region where the DNA is tightly folded. The another term is chromonema. So we have chromomere, we have chromonema. So chromosome consists of two chromatids and each chromatid consists of thread-like coil structure which is known as chromonema. So these are the thread-like structures, they are known as chromonema. It was come into existence by 1912. The chromonemata form the gene-bearing portion of the chromosome. I will let you know during the cell division. Now what is matrix now? Obviously matrix is what basically the mass of achromatic material which is present in the chromosome. So this is known as the matrix. That means inside the pellicle. So the matrix is enclosed in the sheath. Outer layer is known as pellicle. Inside of it we have matrix, we have chromonema, we have chromomeres. Getting my point? So all these things are present in the chromosome. So these all things are present in the chromosome. We have chromatid, which is the like one, two, one like here we have one chromatid and we have two chromatids. Centromere, which is present in the center. Then we have telomere, which are present at the end. Secondary constriction is there. After which we have satellite chromosome, chromonema, chromomere, and matrix. So these is the components or whole components of the chromosome basically. Okay. After it we have composition of chromosomes again. Same thing that Anne Fleming was the one who introduced the chromatin in 1979. The chromatin was classified into two groups on the basis of their dyes. So they are able to, according to the name suggests, they are able to give us color when we apply the dye. So there are two types of colors which we will get. We will get darkly stained region which is known as heterochromatin and lightly stained region which is known as euchromatin. So this is another thing which is there in the chromosomes. So if you are able to see here, the outer ends are known as telomeres. The center is centromere. Now as we discussed before also, darkly stained are known as heterochromatin. So darkly stained are heterochromatin and lightly stained are euchromatin. So they are able to give us two colors. So if you are able to see here, this is a nucleus. This is a nucleus which consists of chromatin. So we are able to get two types of color, light color as well as dark color. Now we have another term which is there in your syllabus. We have karyotype and ideogram. So you have to keep this thing in mind that chromosomes are able to give us color. When we are applying the basic dyes, they will provide us the color. They will provide us the two types of color, dark color as well as light color. Now we have another term which is known as karyotype or ideogram. Now what is karyotype? What is karyotype? The characteristic feature by which a set of chromosomes of species is identified. The characteristic features by which a set of chromosomes of species is identified. Generally, just give me 1 minute please. Hello. हाँ जी मैम, जनिस मैम, कल हमारा एमएसटी का पेपर तो नहीं है बायोलॉजी का? हाँ, कल हमारा तो नहीं है ना, बॉटनी का ही है ना? हाँ हाँ हाँ, ठीक है, बस ठीक है, वो है नहीं क्योंकि मुझे बहुत सारे बच्चे मैसेज कर रहे थे, वो फिर बॉटनी वाले थे। कोई बात नहीं, फिर हम कल मिलते हैं। बस मैंने यही पूछना था, और बस रुचिका मैम को भी मैं यही मैसेज लगा दीजिएगा, बस मैंने पूछना ही था कि कल तो नहीं है कुछ। कल हम मीटिंग करेंगे, फिर हम डिसाइड कर लेंगे टाइम डेट उसकी। ठीक है मैम, ठीक है, ओके मैम, ओके। So I'm really sorry for the interruption. Are you getting my voice? Please let me know. Are you getting my voice? Just 1 minute. Are you getting my voice, Nidhi? My voice is audible. Nidhi, my voice is audible. Please let me know. Okay. Thank you. Deshna, thank you. So yes, this is another term which we have. We have the karyotype. What is basically the karyotype? Yeah. So the characteristic feature by which a set of chromosome of a species is identified. So this is set of chromosomes. The characteristic feature by which a set of chromosome of a species is identified. Generally, karyotype is represented by arranging the chromosomes in descending order of size. If you are able to see here, these are the, according to their size, they are the large one, and when we will move, they are the smallest one. So they are arranged, the chromosomes are arranged in the descending order of their size, keeping their centromeres in the same line. So the centromeres are always placed in the same line. The karyotype of a species can be represented diagrammatically, showing all the morphological features of a chromosome. So they are presented diagrammatically, and then we are able to see all the morphological features of a chromosome. Such a diagram is known as ideogram. So this is basically ideogram, or you can say karyotype. What is basically karyotype? Number 1, the karyotype is the representation when we are arranging the chromosomes in descending order of size. When we are arranging the chromosome in descending order of size, keeping their centromeres in the same line. So chromosome karyotype is basically arrangement of the chromosome, and ideogram is the representation, the diagrammatic representation of that particular karyotype. Getting my point? We have two terms, karyotype and ideogram. Karyotype is so karyotype is the representation in the karyograph is the arrangement of the chromosomes, okay, according to their size, and ideogram is the diagrammatic representation of that particular chromosome. So these two terms are little bit together. These two terms are little bit related together, but you must know the difference. Now there are some special types of chromosomes which are present. We have giant chromosomes, or also known as polytene chromosomes. Let me check here. So we have giant chromosomes. If you are able to see here, they are the very giant. These are the exceptional chromosomes, basically exceptional chromosomes, and these are found in the diphtheria salivary glands. They were first discovered by Balbani in 1882. So these are found in the salivary gland chromosomes. You have to only remember this thing, that they are the giant chromosomes. Their size is 200 times or more than the normal chromosomes. So they are that much, big in size. So this is again the modification, you can say, of the chromosomes, and this is known as polythene. When we have the giant chromosomes, this modification or this mutation is known as polythene. Then we have another type of chromosomes which we are known as lamp brush chromosomes. Let me show you. So these their shape is brush like. So they were first discovered by Fleming, and they are found in the oocytes of sharks. In the oocytes of the shark, they are known as lamp brush chromosomes. Because of their shape, they are known as lamp brush chromosomes. So these are only the chromosomes, if you are able to see. When we are just watching them under the microscope, this is the presentation of them. Isochromosomes, that means they are iso or similar like the original one. So this is a normal chromosome, if you are able to see. But here in isochromosome, we have one short arm and we have one long arm. So in case of isochromosome, one short arm is there, one long arm is there. Then we have some sex chromosomes, like the female has XX chromosome and the male has X and Y chromosome. The Y chromosome is small in size or shape. So this is the Y chromosome and other is the X chromosome. So that is all the things which are there in the chromosome. Now this is the thing, we have cell, we have nucleus. Inside the nucleus we have chromosome, and then we have the DNA and we have the gene. So you have to tell me about everything regarding it, okay? So again they are the same thing. Chromosome number is there. Yeah, so you have to, this is a table which I want to discuss. Like in case of humans, you have to remember we have 46 somatic. In case of somatic, chromosome number is 46 and gametic number is half of it. So similarly, it is happening in every, like in P we have 14, the chromosome number is 14, but the gametic number is 7 only. So the gametic number will be half of the somatic number. Just keep these things in mind. So this is again the centromere. We have chromatids, we have telomere which is at the center, we have the kinetochore along with the centromere. These are again centromere, chromatids. You have to read it by your own self. Okay. Yeah. These are again, yes, this is a diagram which I want all of you to draw. So we have metacentric chromosome in the centromere is in the center. Some metacentric in which one arm is long and one arm is short. Acrocentric when only short arm on the one side and another is too long. Telocentric when there is no arm on the another side. Karyotype is again complete set of chromosomes in a species is known as karyotype. So we have the complete set of a chromosome. The basic number of chromosomes in the somatic cells of an individual or a species is called somatic number. We have discussed this again. So this is ideogram. Ideogram is a representation of or diagrammatic representation of chromosome morphology in a population is known as ideogram. So this is the chromosome morphology. So if you are able to see here, we are just discussing 23 chromosomes. Okay. So these are the 22 are the somatic one and 2 are the, or 1 is the sex cells. So this is the chromosomes, the ideograph of the chromosomes. So you have to read it by your own self, the things I have discussed already. So you have to tell me the difference of gene, DNA and chromosomes. Though this is centromere when in the center, submetacentric, acrocentric and telocentric. So I think that's all for this. These are bending techniques. We will not discuss these bending techniques here. That is all for this chromosomes, and again we have the same chromosome things are there. Everything is discussed quite in detail. I will share the PPT and you have to do it by your own self. Is it clear up to now? Is it clear to all of you? Please let me know. Then we have to start another thing right after it. Is it clear to all of you? Please let me know, because already 1 hour is left. We have done. So I have to start the next lecture. Is it clear up to you, all of you? I will share the PPT, then you will able to get it more. So this is the thing which we have to study in case of chromosome structure, morphology, number, the types of chromosome, karyotype and ideogram. Now the term which I want to discuss is mitosis and meiosis. So up to now, is it clear to all of you? Now I have to discuss mitosis and meiosis. Please, please keep your patience along with you, so that you will be okay. So now I have to discuss some. Oh my God, 151 slides of it are there. Okay, I will try to do it fast. Let me check meiosis also, because these things are quite important. So there is only 21 slides, so I will start with this one only. First I will start with this, then I will go to the high one. Okay. So is it visible to all of you? Please let me know, because I have to give you homework from this cell division. Up to now, is it clear to all of you? And I just request any one of you to please write the questions on somewhere, so that otherwise I will forget also. So you have to type it on the group also. So please write the question somewhere which I ask you in between. So shall we start now with cell division? Please, any one of you, let me know. Shall we start? Shall we start now? Are you feeling bored? Everything is fine? Yeah, thank you, Lopa. Oh, okay, start then. Yeah, thank you all, thank you. So I will start with mitosis and meiosis, which is the cell division. Let's start it. It's a very interesting thing. I hope you are getting. So now we have to discuss cell division. So it is a series of events that takes place in a cell leading to its division or duplication. So when the cell is able, when the cell is complete, now it's a time for the cell to divide, because it is a law of nature. So cell has to divide, or you can say duplicate. So in the cells without a nucleus, the cell cycle occurs by a process termed as binary fission. So binary fission is simple. From one cell we are getting the two cell. That's only the thing. So in the cells with the nucleus, the cell cycle can be divided into interphase, interphase during which the cell grows, accumulate nutrients needed for mitosis and duplicate its DNA during mitosis phase. So we have two phases. Now one thing is there, that means in case of bacteria, in case of prokaryotes, one cell is divided into two cells simply by binary fission. Now in case of eukaryotic cells, the cell has two phases, interphase and mitotic phase. We have to study mitotic phase only. Interphase is the resting stage when the cell is preparing itself. So we have to study mitotic phase. So during which the cell splits itself into two distinct cells, often called daughter cells. And finally the cytokines, where the new cell is completely divided. So from the one cell, so we have two stages, interphase and mitosis. Interphase is the preparatory phase. We will not discuss this, we will discuss mitosis only. So if you are able to see here, this is a structure. We are not able to see chromosomes yet. We will able to see chromosomes only during the mitosis. Is it visible to all of you? Please let me know. Students, is it visible to you? Yes, it's visible. Because the colors are little bit highly contrasted, that is why I asked you. So we have to discuss mitosis. Now what is basically mitosis? What is mitosis? You have to keep two things in mind. In case of prokaryotes, there is binary fission only. From one cell we are getting the two cell, number one. In case of eukaryotic cell, we have two phases. One is interphase, which is the preparatory phase, and the second is mitosis. Now what is mitosis? Mitosis is a process by which a eukaryotic cell separates the chromosomes in its nucleus into two identical cells. Mitosis is a step where we are able to get two cells from a single cell. Okay. So, but one thing we have to keep in mind that when we are making two cells from a single cell, so everything should be distributed equally. Everything which is there in the cell, the organelles and everything, the DNA, it should be distributed equally. So, this is the thing that we have to divide nuclei, cytoplasm, organelles, cell membrane equally. So mitosis and cytokinesis together define the mitotic phase of cell cycle. So mitosis is the basically cell cycle when mother cell divides into two daughter cells which are identical to each other and to their parent cell. So from one cell we are able to get the two cells which are identical to each other. So the number one stage in the mitosis is prophase. We have prophase, metaphase, anaphase, telophase. So this is again your homework. You have to draw the four stages in case of mitosis. I know you all have done this before also in your small classes in plus 1, plus 2 maybe. So you have to draw mitosis, the four stages in the mitosis with colored sketches. So we have prophase, metaphase, anaphase and telophase. What happens in the prophase? What happens in the prophase? In the prophase, the chromosomes coil. So if you are able to see here, the nuclear membrane disintegrates, spindle fibers form. So here there is the formation of the spindle fibers. This is the complete cell and we want to divide this cell. These are the chromosomes. So this is the preparatory phase. The membrane disintegrates, spindle fibers form. In the metaphase, this is a very important stage. The chromosomes, they are aligned. The chromosomes are aligned on a single line. The nuclear membrane is completely gone. So now from here the cell will divide. Because the chromosomes are aligned in a row, there are the spindle fibers which will just attach with the chromosomes. So centromere is there. So the chromosomes, they are aligned in the center. In the anaphase, these chromosomes get separated. So the chromatids separate. These are not chromosomes because they are chromosomes when they are present together. So now the chromatids basically they get separated and the number of chromosomes gets double. So from here we will get two cells from a parent cell. So pushed and pulled toward the opposite poles by the spindle fibers. So spindle fibers, they will tightly pull them apart from the remaining part. So by this we are able to get the two, from one cell we are able to get the two cells. So if you are read here, pushed and pulled toward opposite poles by the spindle fibers. And in the telophase, there will be the formation of the again the nucleus, cell wall and everything will be there, and from the one cell we are getting the two cells. This is a very simple thing in the mitosis. Are you get it? So there are four stages: prophase, metaphase, anaphase and telophase. You have to draw only by the help of colored sketch. You have to draw these four stages. Now what happened in the meiosis? This meiosis is basically little bit more difficult than mitosis. I will try to I will try to explain as much as I can. Otherwise, then we will have to take tomorrow also. If you will not get it today, no issues. Then I will discuss tomorrow also. So in meiosis is a special type of cell division which is necessary for sexual reproduction. So meiosis is for the sexual reproduction in the eukaryotes. The cells produced by meiosis, they are known as gametes or spores. So the gametes are basically sperm and egg cells. So by meiosis we are getting sperm and egg cells. So meiosis is of two types. Meiosis 1, which is the reductional division, and meiosis 2, which is known as the equational division. So we have two types of division which occur in the meiosis. But it is very interesting and very easy. You will get it, no issues at all. So meiosis is generally occur in the sex cells or in the sexual reproduction. It is useful in the sexual reproduction. So meiosis 1 separate the chromosomes. So meiosis 1 is completely similar to mitosis. It is very similar to mitosis. From one cell we are getting two cells. So meiosis 1 separates homologous chromosomes producing two haploid cells. So meiosis 1 is referred as a reductional division. A regular diploid cell contains 46 chromosomes and it is considered 2n. Get it? Simple somatic cell contains 2n, or you can say 46 chromosomes. But in gametic cell, how many chromosomes we have? N chromosomes. That means only 23. So it contains 23 pair of homologous chromosomes. However, after meiosis 1, although the cell contains 46 chromatids, it is only considered as being N with 23 chromosomes. This is because in the anaphase 1, the chromatids will remain together as the spindle fiber pulls the pair toward the pole of the new cell. In meiosis 2, an equational division similar to the mitosis will occur where the chromatids are finally split and creating a total of 4 cells. So let me show you. We have similarly prophase, metaphase, anaphase and telophase. Like in mitosis, we have prophase, metaphase, anaphase and telophase. Similarly here we have prophase 1, metaphase 1, anaphase 1 and telophase 1. Okay. Now in prophase 1 we have 4 stages: leptonema, zygonema, pachynyma, diplonema and diakinesis. So these are the 4 stages in prophase 1. So what is number 1? Leptotene. So leptotene is basically when the chromosomes are getting condensed, they become visible and they are attached to the nuclear envelope. So if you are able to see, they are attaching with the envelope. Zygotene stage, when the chromosomes begin pairing in limited regions. Pachytene stage, this is the important stage. Synaptonemal complex extends along the entire length of the chromosome. Diplotene, chromosomes separate but they are held together by the chiasmata. And diakinesis, when the chromosomes are condensed, detached from the envelope, chiasma remain, all 4 chromatids become visible. So in the diakinesis we are able to get the 4 chromatids. In metaphase 1, definitely these chromosomes will be aligned at the centrioles are there at the opposite pole. So these chromosomes, they are now tightly coiled and condensed as they will remain in the meiosis. In anaphase 1, these will separate. So if you are able to see here, this is the metaphasic plate. So we are able to separate these chromatids in the anaphase 1. Now in the anaphase 2, the homologous chromosome pair complete their migration to the two of poles as a result of the action of the spindle. Now a haploid set of chromosomes is at each pole, with each chromosome still having 2 chromatids. What happened in the prophase 2? From here, we are just continuing just from this diagram. Now the chromosome will duplicate here. Okay. Centrioles duplicate. This occurs by the separation of 2 members of the pair. So again, if you are able to see here, the same now these chromosomes, they will align now. They will again align, and now in the anaphase, they will again separate. So in the telophase, we are getting 4 haploid daughter cells. 1, 2, 3, 4 from this stage. But there is one term which is crossing over, which takes place right now. If you are able to see here, we have light purple with dark purple. We have dark purple with light purple. That means when they are, if you are able to see here, even also this orange has little bit red. This dark purple has little bit purple, because there is a crossing over which takes place here. So when they are present here in the metaphase phase 1, there is a crossing over between the chromosomes. Now what is the need of crossing over? Because there is male cell and there is a female cell. So there is a crossing over for the passing of the characters from both the parents. There will be the passing of the characters to the next generation. So if you are able to see here, before they are fully purple, light purple, fully orange, fully red. But now there is a crossing over which takes place, and when we are in the anaphase stage, so in the metaphase 1 is the very important stage in the life of an individual. Not individual, in the life of a cell basically. Why? Because in the metaphase 1 there is a change of characters. From the father and from the mother there is a exchange of characters. So crossing over takes place right here. So in the next stage there is a separation of the chromosomes, okay, but with the different characters. So they are not similar as the parent cell now. They just cross the character. The chromosomes get crossed. So at the end we will get the chromosomes with different from the original one. So you are getting the orange one with little bit of red one, the red one with little bit of the orange one. So now the characters are exchanged fully. So in the next generation, the characters are from both parents, from mother as well as from the father. We have the characters from both parents. Are you able to get it or not? Please let me know. Have you done mitosis and meiosis before also? I know. So that is why I just keep it very simple. But you have to keep only one thing in mind, that in meiosis there is a exchange of characters. There is a exchange, there is a crossing over which takes place in meiosis. Otherwise everything is same like the mitosis, but there is a exchange of characters only. I hope it is clear to all of you. I am hoping for the best. And yes, that's all for the today's session, and thank you all for your patience. Are you getting me? Everything is fine. Yeah, I will send the slides, no worry. Just be ready with your assignments, the homework. Any one of you please revise the homework again, so that we will able to, any one of you will be have to paste it in the telegram also. Please let me know, have you able to get it? I hope you all have done mitosis before also, so I am not doing it that much in detail. I know you have done it before also. So please, any one of you, please write the syllabus. Yeah, please. Yeah, I will send the slides, no worries. Just give me some time. I will send it definitely. So please write the homework which you all have to do. Please type it fastly. What is the thing which you have to do? Yeah, very good, Bhavni. What is the difference between gene, chromosomes, and DNA? The number one question. What is on number two? Please. I just already told you before to write it somewhere because I forget then. Yeah, you have to type the question. Please, what are the another questions which I gave you? You have to make the diagrams of, yeah, acrocentric, metacentric, the chromosomes. You have to make 4 types of chromosomes. Yes, very good. And then you have to draw mitosis as well as meiosis, the 4 stages of mitosis as well as meiosis, but with the help of sketch. Okay, so you have to show me the crossing over in case of meiosis 1, in case of metaphase 1. When there is a crossing over, there is a change of characters, so you have to show me the crossing over. So for it you have to use different sketches or different colors. So is it clear to all of you? Please let me know. Yeah. I forget, Bhavni. Thank you. One more question is why mitochondria is semi-autonomous in nature? Why mitochondria is semi-autonomous in nature? Thank you, Snadhan. Also, so these are the questions you have to you have to write in your notebook or on the MS Word as your wish. But you have to send it in the group, in the Telegram group. Then I will check it. So I just want to thank all of you for your patience. You are here continuously with me and you are just listening to me with full concentration. I just want to thank all of you. So yes, please write down the homework and any one of you please type it in the telegram also and pin it so that all of you will be able to answer the questions. I think that's all for today. Thank you all for your patience, for everything. And yes, we have done two things today. The chromosomes which are left yesterday along with mitosis and meiosis. So tomorrow we will meet again. And till then, take care of yourself. Good night from my side. Take care of yourself, all of you. So and also try to do the homework as soon as possible. But don't be just relax. And when you will be free, then you have to do your homework. Okay. So thanks all of you. Thanks Nidhi, Pooja, Bhavni, Snadhan, Priyasha. I thank all of you and good night.